So we are now going through the same slides, virtually the same slides, and try to attempt a short review for particularly for students who need a little bit more of help navigating through the material, which is dense, as we all know, because anatomy is a very dense discipline. So here we have the four primary tissues, and a slide like this, like you heard me say many times, may be a very important slide to begin with, but also to end with after your studies, in the sense that it contains a lot of information that seems very basic, but is essentially information that you are able to understand better once you know many more of the details that you cover throughout the lecture. So you have here essentially four types of tissues. So in a slide like this, you do need to spend enough time, and I always tell students to spend enough time, meaning that it's important for you to allocate the right amount of time to make sure you understand all the information that is contained in the slides. A slide like this contains, for example, a picture that you could analyze. So we have four types of tissues: epithelial, connective, nervous, and muscle tissue. So it's relatively easy to focus on these three: the epithelial, the muscle, and the nervous, and leave the connective tissue as this set of tissues that are very heterogeneous and that we would need to know much more about it. But the epithelial, so you see, for example, here, if you already went through the slides, you would know that this is a squamous epithelium, that you see the cells flattened on top. But the epithelial tissues are tissues for lining. So every time we think about something being lined, so either from the outside or from the inside, we think about epithelial tissue. We will talk about a specific type of epithelial tissue that are the tissue that is associated with glands, and that is also an organization that consists in great part of epithelial tissue. The muscle tissue is the tissue that we all understand what it is, so produces movement. But the novelty for a student who never learned a lot about tissue is to know that there are different types of tissue: the skeletal, associated with the skeleton, the cardiac, associated with the heart, and the smooth muscle, associated with the viscera or the internal organs besides the heart. The nervous tissue is relatively easy too. It comprises of tissue that is important in terms of the nervous system, so we can have classic definitions such as receiving signals or passing signals or conducting electrical signals or carrying information. And again, the connective tissue is connecting, but it's a big bag full of tissues. So you see, for example, when it says stores energy, we're talking about the fat tissue, the adipose tissue, providing structural support. We're thinking about the cartilage and the bone. Filling internal spaces, so you talk about the connecting aspect of it. But of course, if you think about the blood as a part of a connective tissue, we can also think about other functions. So again, this is a slide that you could come back to it. So the epithelial tissue is composed of cells that are particular in terms of their anatomy, and you have to have a certain understanding of the terminology of these cells. So spend some time here thinking about what the cells, the epithelial cells, is seen as something that has a bottom and a top, and so it's sitting or resting somewhere, and like the skeleton, you can produce terms to describe this. So I'm not going to go over everything. I just wanted to basically cover otherwise this becomes a second lecture, but pay attention to what is basal and what is apical, and then think about a picture like this again, showing you several different of these modifications that we'll talk later. See, epithelial tissues don't have vessels, so avascularity. So that's one reason the epithelial tissues are very constantly needing division. These are the functions of epithelial tissue, so go over them slowly, but they should be intuitive. They shouldn't be, they make sense. We are not used to thinking about certain functions, but it's the same thing as me asking you, what are the functions of your walls? Or what is the function of paint on the wall? If you think about why do we paint walls? And you know, you can think, oh, it's beautiful, which is one aspect, but also, you know, paint oftentimes protects. Protects from what? From abrasion, from destruction, from allowing certain things to pass. So we have to, in anatomy, we spend some time thinking about functions that we may take for granted or not even notice in other parts of our lives. So the specializations of the cells, you need to know what are they. Again, they should make very much sense. Pay attention to these three components that look like similar at the microscopic, or at least at the diagram, but cilia and stereocilia and microvilli are different things. So here is where you're going to start allocating, so associating, putting in the same box certain things. So microvilli, we'll think about them when we think about the intestinal epithelium and a way of absorbing more. So it's perfectly okay for you to fix on this part, but you see the slide says fixate on this part, and the slide says but there's also urinary cells. So it is okay that you forget that is also in the urinary cells, but you focus on the major aspect. Microvilli, think about intestinal cells, think about absorption. Stereocilia, you think about, for example, cilia that move, but with a certain purpose, and that the movement may represent certain signals, such as, for example, the cilia that we have in the cells in the inner ear. I hesitate in telling you the name of the cells, but it doesn't matter right now to tell you all the names. Then the other cilia, they just move substances around, and these, think about any type. You have movement of mucus, for example, such as, you know, respiratory tract. So you see here that I'm going very slowly, but just to help you understand how you should be studying this after the first round of slides. So I am trying to associate, I'm trying to make meaning of it. Meanwhile, the material adds up and you have more and more, and you may think, oh, this is more for me to memorize. So then you go over, pay attention to the pictures. They are for particular, for even for everyone they are important, but there are some of us are very visual in our learning, so sometimes we memorize that better. The epithelium, one way of looking at it is seeing like tiles, and so the tiles need to be attached to one another. So understand these terms, understand what are intercellular connections, and then we'll go over the types. The attachment to the base, which is the basal lamina, and a lot of times we confuse basement membrane with basal lamina, but it's important to understand that basal lamina is part of the basement membrane. Here are the types of the microscope, the parts of the microscope. So you see here the different types of microscope. In the intercellular attachment, we are going now for a different levels. So we covered the basics, and now we go to a level up. So we go into a detail. The first thing is to understand the cells need to connect with one another, and now is understanding what kind of connections. So here your goal should know the names and the meaning, but also the location of them. So again, you don't know location if you don't have anatomical landmarks or anatomical points of reference. So again, you imagine the epithelial cell as sitting on top, so having a top and the bottom, so the apical and the basal, and then you have to know the different types of intercellular connections through the perspective, through that anatomical perspective. So it's not only just understanding that their structure, but also where they sit. And so here it's we're going to go over, you're going to go over them, each one of them, and knowing everything that is on this caption is already enough. But the names also, again, I'm not going to make this into a lecture, otherwise it's going to be too repetitive. But tight, see the word tight, if it tells you something about what the cells are. Gap, so the word gap is allowing you to understand there are passage, so there are gaps, there are passages, and then there are the spot desmosomes and the hemidesmosomes. Pay attention because the hemidesmosome and the spot desmosome are in different locations, so you do need to know the locations. And this is just a summary for you to, again, I like to include pictures that go back to the bigger picture to understand if you can identify them and create your own summary. So now we go to a level 2, but in terms of epithelial tissue. So the basics, there will be a repetition. Again, I explained this in lecture that you have to understand what is simple versus stratified, and when and then you have to also understand what are the cells made of, or what type of cells. The cells are classified based on where you see the cells better. So think about seeing the cells where they have more freedom. When they have more freedom, are they flattened, or they look like a square, or they look like a column? This may be tricky, but it's important to understand in the same row, you don't have different types of cells. So in the same row, you have the same type of cells. So it's possible through a cut, you get doubts, but if you look at more, you see the majority of them look what? A square, they look flattened, or they look like a cube. When I say the same degree of where you have to analyze it, so the lumen is the hollow part of a tube, so here's where the cells have freedom, and you see they are square. If you're looking at the basal part, you could think, oh, they are cubes, but actually they are columns. So columnar, cuboidal, and squamous. And again, these are for you to go over them. Again, the squamous, now your goal should understand where do you find each of these tissues, identify them for lab, but also where do I find them? And there's particular names. For example, the simple squamous has names depending where it fits. For example, the mesothelium lining surfaces, body cavities, and the endothelium lining the blood vessels and the heart. The function should not be something that you get too locked into, because functions should make sense, and it's also not something that we could argue. There's a certain level of freedom to see functions of things, whether they make more sense or less sense. Sometimes that's a little bit debatable. But again, it's like the same question I asked you: what's the purpose of the tiles on the floor? What's the purpose of paint on the walls? So I wouldn't be too much worried about that. Then stratified squamous. Stratified implies that there's more than one layer, so you think about a little bit possibilities of the different layers being somewhat different. So you think, for example, of the keratin being accumulated in the outer layer, such as in the skin, or the mucosa, the insides, the lining of our inside, like our mouth, our stomach, and so on and so forth, is non-keratinized. The cuboidal exists in many areas. So here is trying to find. Normally, we give the example of the kidney tubules, but there are a variety. And then the stratified cuboidal, so more layers. So think about, for example, ducts where you have more than one layer. So the reason, again, you see layers, or the way you see layers is looking at the nuclei, plural for nucleus. So if you see several layers, again, there's no mistakes. So if you say, oh, wait a second, but here there's only one single and here's two, well, what you look at the others and you reveal that there's actually more cases in which there are two layers. So the cuts don't always show you the nucleus, and so you have to pay attention to the everything, but it's almost looking at types of trees. So they are all the same. So if you find in one and don't find in the other, chances are that just the cut doesn't show it here, but shows it here. The columnar, again, what would be your example? The simpler columnar, think about the epithelium that is in the intestine, for example, those are the gastrointestinal tract. And then think about simple columnar as having modifications. So you can have simple columnar modifications that have cilia. Cilia, you automatically think about, for example, in this case, the fallopian tubes, moving secretions, and the simple columnar that don't have cilia are the ones that have microvilli. When we say have or don't have, this is not a 1 or 0. You see that there are spots in which you have, spots in which you don't have, and spots in which you do have. So the microvilli, you think about the intestine and the proximal renal tubules. This is another example. I bring you a lot of examples because the examples are from different textbooks. There may be a little bit more added information. This is where a student needs to be very cautious in not being so hooked onto a lot of details. You're going to have an exam that includes a lot of questions, so there's no time to go on particularities. So focus on major examples. Don't focus so much on tiny examples. Okay, so if there is a little discrepancy, or one slide says more than the other, that's okay. It's because one textbook is more detailed and others is less detailed. But you focus on the most important ones. Think about this. It's okay to forget a detail, but don't forget the major picture or the major facts. Pseudo-stratified, so pseudo, the word false, so it's something that looks stratified, but then when it's stretched out, for example, or when you look at it from a different perspective, you see that actually just one layer. This is the epithelium. The pseudo-stratified ciliated columnar epithelium is pseudo in the sense because it looks like multiple layers, but it is in fact is one, because all these cells are attached to the same basement membrane. And think about the respiratory tract. You see here, for example, the goblet cells. So pay attention. I am going fast because, again, this is just to alert to and help you focus on certain things, but you do have to go slowly. The stratified columnar is similar to the pseudo-stratified, but it has more layers. And in this case, you find the pharynx, in the epiglottis, in the anus, the salivary glands, for example. So, for example, you think about the urethra, but you see the different types of slides. Say it's small areas in the pharynx, so meaning that you don't find it all over. And here in the textbook, the different textbook even says that it is in the urethra and not much more. The transitional epithelium is epithelium that depends on the state of the organ, whether the cells are flattened or stretched. When the organ is stretched, the cells are look more flat, whereas if the cells are not stretched, they look like more cuboidal. That's why it's called transitional. This is talk about the bladder, the urinary bladder, but may also appear in the renal pelvis, in the ureters. So these names, pelvis, ureters, and all that, these are names that you may not know what they are, and but you definitely need to start knowing. So don't let these terms go like, what is he talking about? What does he mean? You have to search for it online, and then when we cover that, it's going to be more clear. So this would be where you stop and you try to classify. I would add here the example, okay, the major example of each one of them. The glands, the classification of the glands is a bit tricky because it may be very complicated, so don't let yourself go overboard, but there are main classifications. This slide contains the high yield, so the difference between exocrine and endocrine, and the difference about the type of secretions. Are they more watery? Are they more mucus, more thick, more sugary, which is have the glycoprotein, or they are more mixed? The endocrine secrete to the blood or lymph, and therefore they are hormones. The classification is important to a certain extent. So think about the unicellular glands. We talk about the goblet cells, the secretory sheets like in the stomach, and then the structure of the exocrine when it has more than a simple one cell or a simple layer. You have there is a duct and the secretory part. So understand this anatomy is going to be very important. And so you can then classify it whether the cell is this based on the secretory portion, just the secretory portion, so it's focused on the pink. You have whether there's just a tube, whereas they make a alveolus or acinar gland, or if you have tubes and these bags, they're tubular alveolar. Then if the duct itself, it's the bluish part, can also be simple or complex. Complex branching or called compound. This is a slide that may seem very trivial, but is a slide with a lot of information, which is the type of secretion. This is high yield things, particularly for future courses. So how do I secrete? Do I secrete by releasing vesicles, which is the eccrine or merocrine? Do I secrete by shedding the upper portion, or do I secrete by bursting the entire cell, the holocrine? So you would pause where you feel is fit, and then connective tissues are complicated. So the connective tissues, we're very happy if you first of all know that it includes a lot of tissues, and the anatomist, to try to make sense of it, is going to try to make common denominators, so common classifications. So all the connective tissues have a matrix and have specialized cells. The matrix is what happens outside of the cells and the cells. So you have cells and everything else is the matrix. So the functions don't really matter because it will depend on the type of tissue, and they should make sense. So the classification is a bit tricky, you know. Again, you have to go with this on your own, but proper means that is the actual connective tissue that we think about, the connective tissue that's just binding. And the question is it loose or is it dense? Loose because the fibers are loosely packed, or dense because the fibers are densely packed. Then there's fluid connective tissue, and we think about the blood and the lymph. And then there's supportive connective tissue, supporting connective tissue, which is the cartilage and the bone. For all of this, you now, more important than knowing all the details, is to start knowing the certain names. So you start with a connective tissue proper and having names of cells. So you do have to know these names, you do have to know them and make sense of them. To make sense of them, you have to focus on slow down this. Then the type of fibers, are they collagen, are they reticular, or are they elastic? And the type of connective tissues proper that you have. So you have the loose connective tissues, the dense, and what types of them there are. And here this becomes like almost level 3. So you are going to an upper level where we having going into more detail. So focus on where you find this and the main characteristics. Of course, if you think about the don't lose track, it's very easy to lose track, and that's why I like this slide. Connective tissue proper, loose, areolar. Adipose tissue is a high yield tissue, of course, so you have to know the difference between white and brown fat and the classification. Look at the cells so filled with fat that the nucleus is pushed to the side. The reticular tissue is a very important tissue to form the framework, or sometimes called the stroma of an organ. And then, so think about solid organs, organs that have that you could cut with a knife and make a slice, and they have a reticular. They have an organization that is internally. Then the dense, think about something that is heavily packed, and this you're going to think about, for example, tendons, aponeurosis, or elastic tissue. Again, if all these are words that you don't know, you have to try to get to know them. And the definitions show up here. These are terms that are starting to be introduced to you, but then when we cover them during the anatomy of the muscles, you will understand what they mean better. The dense irregular, so think about sheaths, so things that cover muscles and nerves. And then elastic tissues, think about things that have elasticity, such as, for example, vessels. And the fluid, again, these are the simple ones, the blood and the lymph. In the cartilage and the bone, we're going to cover this more, but you start having the word cartilage associated with chondro and bone associated with osteo. And the different types of cartilage, which you absolutely need to know because we're going to even cover that in more detail. So, and this is the anatomy of it in terms of how they look like. The hyaline fibrocartilage or elastic cartilage. Your goal should be to understand what it looks like, so identify, but also what is the main structure in the body where you can find this hyaline cartilage. So pay very close attention to the location and where it is. So these are questions that oftentimes or these are things that topics that oftentimes can render a lot of questions, and students may struggle because they are very much forgot these things. So the hyaline cartilage is the cartilage that we normally think about. Cartilage, like if you buy a piece of meat and someone told you with bone and joints, you think about cartilage, and so that cartilage is that. If you break a joint, you see that is the kind of cartilage you find there. So this is the hyaline cartilage. So again, pictures, they include a lot of details. They have a joint here. This is the shoulder joint. And the hyaline, why is hyaline? Again, because the hyaline, the matrix does not stain, so it's more glassy. Another example here is, so when we talk about the ribs and how the ribs connect to the sternum, ribs are these bones, the sternum is this bone, we talk about hyaline cartilage. So think about joints, think about the costal, the cartilage that connect the ribs to the sternum, it's called costal cartilages. And now we think about something that you may not immediately know that is cartilage. The elastic cartilage is the cartilage that you find, for example, in the ear. So think about the ear or the epiglottis, which is the part of the throat that connects to the trachea. And then you have the fibrous cartilage, which is the cartilage that are more for pads. Where do you have pads? Well, you have pads in the knees, which are the menisci. You have pads between the vertebra, which are the discs, and you have pads between the pelvic bones, which is the pubic symphysis. Again, once you do a reading of this, these reviews make more sense. If you otherwise, this becomes very complicated. The bone, we're going to cover it much more, but appreciate the names: osteoclasts, osteocytes, osteoblasts. They are three different cells that should make sense to you from the beginning. Osteoblasts form bone. I'm not sure how mnemonics you can use, but the B as in bone formation. Clasts destroy the bone, and the osteocytes are the mature bone. Summaries like this are interesting for you to see if you can organize. Don't get too locked into details, because again, there will be more places for you to know these details. A very level up is knowing the types of collagen. In a basic anatomy course, it's difficult to find room to ask a lot of questions about this, but it's important to understand there are different types of collagen. Membranes is a concept that you'll hear a lot, and up to here you only think about cell membranes, but again, think about the membrane from an anatomical perspective is an epithelium plus connective tissue, and the difference between mucus and serous, cutaneous, and synovial. So those are important for you to know. And this is the fascia, which is the different types of layers. Again, if you're not a vegetarian and if you don't mind cutting a little meat, it may be important for you to analyze the meat even when you buy it. Particularly, some meats are some type of meats. It's easier for you to see it, of course, but even if you're just at the grocery store, just trying to look at the meat can be interesting. Of course, you have the lab where you dissect too. The muscle we're going to cover muscle many times. We're going to cover the muscle when we talk about the skeletal muscle, the heart muscle, the smooth muscle, but you are invited here already to know all the differences. So one thing is to understand where do you find these muscles and the names. Understand the names of the cells, the muscle cells, may have different parts, like the sarcoplasm is the cytoplasm, the sarcolemma is the cell membrane. And then understand why we call skeletal and identify them anatomically. Okay, so think about criteria: multiple nucleated, and the striped appearance. The cardiac muscle, it only has one nucleus. It has a certain stripes, but they are they are separated by intercalated discs. So sometimes they call striated involuntary muscle, and they are branched. They form these Ys. This even shows you a little bit more. And then the smooth muscle, which is for involuntary contraction. Think about anything in the body, inside of the body that needs contraction, that's not the heart, has smooth muscle. The nervous tissues, the neurons, and the glia. So I hope you appreciate this review. Again, the point is not to go over everything, but to help you study or actually review using the slides and to see if you are headed in the right direction.